what is, it's a good, it adds a different flavor to the talk. Yeah. So what is Nemp? This seems a little bit falling off the things, but Nemp is a 5-transmembrane protein that, is there any way to lower this? Catch the bottom of my book. So it's a 5-transmembrane protein, and it's found in all metazoans. And essentially, up until when we started studying this protein, thanks, Denise.
I don't think it's going.
I think it's only the adjustment. It looks like it's not.
It's, I can,
Sorry.
no problem. And so essentially nobody studied it in vivo. And so that was really interesting to us, trying to understand what this protein did. And a little foreshadowing is if we looked in human genome databases, what we could see is variants in this protein in humans were associated with premature menopause. So that was pretty much all we had. So I'm not going to get into the long story of how we got into this, but essentially we used Drosophila as a rapid-ish system to study what this protein, Nemp1 or CG9723, did in flies. And what we saw was when we made null alleles, so completely null, we replaced the gene with the white eye color. The animals were partially lethal in the pupal case, but all the ones that came out looked normal from the outside, but they were completely sterile. And the other sort of interesting thing is when we looked in lots of tissues, the eyes, the brain, et cetera, it's ubiquitously expressed. So it wasn't obvious at first why that would be. But when we did a little dissections, it was pretty clear why these animals were sterile, because they had almost no ovaries or testes. So this is the Drosophila testes. These are the same magnifications and wild type, and this is mutants. So you can see there's just essentially the little tip is left in the mutants. And this is one Drosophila ovary, and these are 2 ovaries. So it was just a massive effect, and so not surprising that it was sterile. So obviously you worry that your protein, that you've made this mutant, and is it really due to, are your phenotypes really due to mutations in the gene you've made or something else? So we rescued it with BACs, we rescued it with Nemp1 cDNAs, Nemp cDNAs, and you can see, from a tiny, almost not even visible at all on this screen, testes, we got pretty full rescue. So we felt good that we were following the right gene and the right phenotype. And kind of excitingly, when we took human Nemp1 and we put them into flies, we found that they were able to largely, but not fully, rescue viability. So we had, most of them came out, and they also could rescue fertility. So it suggests definitely that this was a conserved protein and pathway. So more about Nemp. We made antibodies to Nemp. We did super resolution microscopy with Nemp. You can't see off the bottom of the screen with the Peltier lab, and we could see that it was an internuclear membrane protein. Okay, so it's a ubiquitously expressed internuclear membrane protein. So we wanted to know where in the germline is it needed, or is it needed in the germline? And so we used RNAi to knock it down. And so this is again like a wild type testes. And if we use a germline specific GAL4 to knock down Nemp, you can see we substantially, but not fully, phenocopy the mutant phenotype. And similarly, in the ovaries, if we knock down Nemp, you can see a dramatic loss of ovarian structure. So this is what Melissa alluded to. I'm not going to. Okay. Okay. Yeah, maybe.
Nope.
No. Okay. We'll continue and see what happens. Okay. So obviously, since human Nemp1 could rescue Drosophila Nemp, we were curious, could we look at model organisms where we could really test this, because we can't in humans? And so we teamed up with Tim Shadle at WashU, and Ariz in his lab knocked out, made 5 different alleles of C. elegans Nemp, and found that if he looked at root size, or maybe I'll just look at percentage of sterility, you could get very substantial loss of fertility, not complete, but a substantial defect in fertility and C. elegans. And so working our way up the ladder, we teamed up with Brian Ceruna in Toronto, and this was actually ahead of the worm work. Vertebrates have two Nemp genes, Nemp1 and Nemp2. And what Brian and Curtis in his lab found was that, if you remove Nemp1 or Nemp2, so if you remove Nemp1 or Nemp2 in fish, there was no phenotype that we could pick up, but double mutants, Nemp1 and Nemp2 mutants, had a substantial loss of fertility in both males and females. I feel like this is sort of a game show here right now.
Sorry, guys, like, I'm not sure what happened here. Apologies.
It's okay, no problem. So essentially, I'll just focus on the females. Nemp1 to double heterozygous, like wild type, will continue laying eggs and being fertile for many, many, many months and years. But if you look at Nemp1, Nemp2 double mutants, this is code-colored for brood size. After just a few weeks, you've substantially lost fertility. Okay. So in my lab, we also work on mice. So there's two Nemps in mice as well. We knocked out Nemp1 in mice, whole body knockout. We just took out exon 3. These are breeding trials. And what you can see here is in wild type, these are color coded. You can see that they reproducibly give substantial litters for the 6-month breeding or 4 to 6 month breeding trial we did. Heterozygous are still fertile, but Nemp1 mutants, do you have this? Nemp1 mutants are almost completely sterile. So over a period of 3 years, we've had maybe 4 or 5 litters of 1 or 2 pups. So it had a really, really strong effect. Thank you. So, okay, that's great. It says Nemp's important. It's important in the germline. It has a conservable fertility, but how's it working? So then we started doing proteomics. So it's a 5 transmembrane internuclear membrane protein. Biochemistry is quite difficult. So we used a bio-ID approach to find the proteins it was close to in HEC293 cells. These were done with human proteins, and the human protein was known as TMEM194. And what we could see is we got a very distinct fingerprint of proteins, but what you can notice, or I hope you can notice, or at least believe me, that the most abundant proteins that we got were a subset of nuclear envelope proteins called LEM domain proteins, things like MRAM, which are so. Okay, so what are LEM domain proteins? So they're transmembrane proteins. They interact with lamins. They're essential for nuclear integrity. And mutations in lamins or emerin or other LEM domain proteins give rise to laminopathies. So these are like progeria, muscular dystrophies. So we validated by co-IP that that these interactions were real and not just a proximity interaction. They form biochemical complexes. So what you can see here is we also did AP mass spec studies, and emerin or Drosophila odorphin was a top hit, and we could validate both. We could validate that this interaction could be was present. Okay, and that was interesting because loss of emerin gives some phenotypes that looked a lot like Nemp mutants. So you could see here, this is a picture from Pam Geyer's lab showing that emerin mutants have a reduction of vasopositive stem cells. And we went back and looked at our Nemp mutants in the pupal ovaries, and what we could see is we also lost the emerin, the vasogerm cells. Okay, so that was interesting, but we were sort of interested in the laminopathy questions and trying to understand how nuclear structure might be affected. And what we noticed in looking at the mutants, these are EMs of germ cells from wild type and Nemp mutants. We used to call it SPEG for sperm and egg yolks. Sorry for that title still. In wild type germ cells, the nucleus is fairly regularly shaped. But in Nemp mutants, what you can see is it becomes like crazily involuted. So that's an EM picture. We all have seen a single EM picture. So I was particularly interested in seeing how robust this phenotype was. And the answer is it's super robust and very early in germ cell development in the testes. So what you're seeing here this is just a single testes with consecutive confocal sections, just working their way through, stained with laminin. And what you can see, and so these are the germ cells. And if you look, you can see some problems occasionally, or problems, little blips on the nuclear envelope of wild type germ cells. But what was super clear is when you look at the Nemp mutants, the hub looked pretty good, but as soon as these cells started going mostly into the transit amplifying stage, look everywhere. We could see these really radical involutions. And so this is actually the first defect we could see in these mutants. Okay, so we were curious if these nuclear envelope defects were restricted to the Drosophila germline, or did we see them more generally as a conserved to humans? So we used knockdown in human tissue culture cells. And so what you can see is the relative level of knockdown, and this shows the nuclear shape. And what you can see is as we got more and more knockdown, the nuclear envelopes got so strange looking. Okay. So that was interesting. We didn't see that in every Drosophila cell, so this is still a question. So we went ahead and looked at a ton of different cell lines. And what we saw overall, and more than I'm showing here, is when we knocked down Nemp in cell lines, in mammalian cell lines, we could see that cells didn't do well, okay? Normally in wild type, you would expect to see the cells to increase in the number and proliferate. When we knocked down Nemp1 and Nemp2, that's M194A and B, we could see in all these different cell lines, we were losing cells. Except we saw one cell, so that's kind of hard because a dying cell, we're working on them now, but it's really hard to get into mechanisms there. So we particularly focused on this one cell line, SHIP77 cells, which are semi-adherent cell lines, so they're not adhering strongly to the surface, and they seem to not mind as much loss of Nemp. So we thought maybe this would be a tool we could follow them up a bit more. And so at that time, we started collaborating with a physicist, Zhu Sun, and his student, Qian Wang, at University of Toronto. And they had developed an atomic force microscope, which they were really excited about because what they had done, instead of using a normal probe, they had used a focused ion beam to make it into a little needle. And so this little needle, if in wild type, as it punctures the plasma membrane, you can see some resistance. And then as it punctures the nuclear envelope, you see again, and actually if it goes into the chromatin, it kind of goes off scale. But in the Nemp1-2 knockouts, so my lab used CRISPR to knock out both Nemp1 and Nemp2 in these SHIP77 cell lines. And what Qian saw was that the plasma membrane resistance seemed pretty normal, but the nuclear envelope seemed to have gotten soft. Consistent with the EM's data or at least the interpretation we had of the EM data that was floppy. And so Yanit, my lab, and Qian did this a bunch of times, both on, sorry, you really can't see. These are Nemp1-2 knockouts. These are just Nemp1 knockouts. And so they could calculate the apparent Young's modulus, and you could definitely see that these cells, the nuclear envelope of these cells had gotten much stiffer, and it seemed to be primarily due to Nemp1. Right? Nemp2 might have a contribution, but it seems small. So then we had shown a biochemical complex of Nemp1 and Emerin, and we were wondering, is Emerin part of this? We had seen this phenocopy, this biochemical complex. And we used SHIP77 cells again and knocked down Emerin, and we could see that, in fact, just like Nemp, it got softer. And then we were particularly interested to see if they were working together. And so what we did was we took Nemp1 knockout cells, and you can see this is with a control SI, they're softer, as I showed you before. But if we knock down Emerin in the Nemp1-2 double mutant cells, they're just as soft. And so we think this argues that they're probably acting as a complex together to support the nuclear envelope. Okay, so at that point, we have this model that Nemp1 interacts with many LEM domain proteins, such as Emmerich and Lapp, to support the nuclear envelope and maintain the germline. And I told you that Nemp1 function is conserved. So we were very excited to try to understand more on a cell biological level what's going on. And so it was actually a little bit easier to do. I mean, the genetics is easier in the fly, but in the cell biology is sometimes easier in the mouse. So we generated antibodies to Nemp1. This is an entire mouse ovary stitched together and stained with Nemp1. And you can see these little bright circles on the corners, or on the edges in the cortex. That's Nemp1. And so you can trust our antibody. Here's wild type, here's mutant. You can see it goes away to mutant. And it's expressed very strongly in the germ cell. So you can see the fossa. Okay, so oocytes start as a primordial reserve that sits at the outside of the ovary cortex. And then certain oocytes are selected to start growing, to become a primary follicle, secondary follicle, an antral follicle, and then they get ovulated, okay? So we looked, and you can just define these different types of oocytes by histology. And so what you can see is that by histology, these are wild type, het, and mutant, looking at primordials, primaries, and secondaries. And what you can see is in the Nemp mutants, so Nemp is really strongly expressed in those primordial oocytes, we go from, we have a substantial loss of the primordial oocytes, right? So the ovarian reserve is largely lost in the Nemp1. So how are we trying to put this together? So I was excited to read that there's actually a whole bunch of literature on mechanical stiffness and fertility in humans. And what people have discovered is that there's a gradient of mechanical stiffness and collagen cross-linking in the mammalian ovary. And what is known is that the cortex is very cross-linked and stiff, and then the medulla is softer. Okay. And so we had noticed, as I told you before, that Nemp1 is highly expressed here. And so we're putting together with our AFM data and our and those analyses that are a hypothesis. And then we also noticed, that Emmerich is also highly expressed. The data isn't as pretty, but it is clearly expressed more strongly in the primordials than later on. So mechanical stiffness by the nuclear envelope is generally thought to be supported by lamin AC. And so you would have thought that lamin AC would be high in the cortex, but in actually what we see is there's almost no lamin AC in the cortex. And so at this point, we hypothesized that Nemp and Emmerich together are working to support the mechanical structure of the nuclear envelope in the face of this high mechanical stiffness. And maybe there's something about this complex that's important for germ cells, maybe meiosis or something like that, but that lamin AC, which normally supports the nuclear envelope, can't. So how do we test that? So we hypothesized that the absence of Nemp leads to loss of oocytes due to cortical stiffness. And so we wondered if a softer cortex could rescue these mutants. So how does collagen get cross-linked? It turns out that at least one of the aspects is that the collagens get cross-linked by enzymes that are known as lysyl oxidases. Okay? And so we wondered if we took away lysyl oxidases, what would happen? That's what it just says, right? And so this is super exciting because it's still such preliminary data. It's actually on this graph. But so locked homozygous die early, so we couldn't do that. But we were able to cross wild type males to Nemp1 mutant females that lacked one copy of lysyl oxidase. And what we now see, so what it should show you is number of pups per litter. But basically, I already told you that you basically, so rarely get pups over years, and we've been breeding these for 3 years, and yet 1, 2, 3, we have 6 going right there. 6 out of 10 animals have now given us litters. They're very small litters, but they are. Sometimes we have 1, 2, 4. So I think this is pretty remarkable, and definitely I think suggests that you can't rescue the phenotype by reducing cross-linking. Okay, so how do I put that together? How do we put that together with the fact that loss of Nemp, this is probably maybe more cells than we showed before, causes, I don't know where that circle's in. You lose cells if you plate them on tissue culture plates. And so when you start reading about mechanical stiffnesses and cross-linking, what you realize, this is from a recent Nature review, is that we're actually quite soft, right? Substrates, maybe we're softer than we should be. So when you take things on tissue culture plastic, you're getting into a really stiff environment. And so we hypothesized maybe that the reason that animals, so because it's weird, right? I just told you that we can have live fish, flies, worms, mice, whatever, without Nemp, but we can't live with cells. And so first of all, this is showing that we reduce proliferation, but and I apologize, this isn't yet, but we've done this experiment like 8 times now. So we've started a new collaboration with Anit Pathak at WashU, who's a physicist, and his student Hannah has been generating these substrates that are different stiffnesses for us to plate the Nemp or knockout cells. And basically, this is 10 kilopascals, 30 kilopascals, 120 kilopascals, and if you plate wild type cells, they basically are okay. But if you knock down Nemp, what you see is in 10 and 30 kilopascals are living. So first of all, that's amazing, because we've been trying to plate these cells and trying to study them on tissue culture plastic, and they're always dying on us, and that's a challenge. But if you plate them on, so now that they're living, that's great, 120 kilopascals, now they're not. So we think that this is at least part of the way that Nemp is working. Okay, so it's a conserved internuclear membrane, it binds Lem domains, it supports mechanical stiffness. Okay, so I told you that you lose the primordial reserve. So we're going back to the mouse to understand the biology a bit more. There's other defects that would be fun to talk to Melissa about. So for example, if you take the few oocytes that remain and you allow them to get fertilized, what you can see is they'll start development, but basically they stop around the 2-cell stage. Okay, and suggesting loss of developmental competence. So that made us decide to go back and look more carefully at the oocytes, at their chromatin structure. So this is showing you primordial oocytes, which I told you before, and then they become activated. They really grow huge. During these changes, they also undergo large-scale chromatin remodeling. And it's a really crazily dramatic thing. So this is an early oocyte in a non-surrounded nucleolus structure. And then it becomes so compact that when you do her staining, you don't even see it. Okay, and they're called non-surrounded, partially surrounded, and surrounded nucleolus stages. And that's just another picture. Actually, the words aren't off, so that's good. Okay, so now we went and looked in the Nemp mutants, and we did ovary sections. And what you can see is what really was shocking to us. So this is a wild type. These are 2 oocytes at the same stage, the preovulatory follicle oocytes at P30 females. And if you do sections and you stain with DNA dyes or histone modifications, you can see this super crazy wrapped structure that the nucleus gets in this oocyte that's almost ready to go forward. But in a Nemp mutant, which is at a comparable stage, what we can see is it hasn't achieved that chromatin remodeling, or its chromatin is disrupted, decompacted to us. You can also ovulate the oocytes and do their staining, and what you can see here is over here, this is the wild type, and you can see that, essentially the same. The DAPI staining seems to be really decondensed. In fact, we got the impression from, if you look at those last pictures, you can almost get the feeling that, is the nucleus bigger? So we wondered, it's not as much bigger as you would think, but it actually is. So if you stain with lamin and you measure the diameter, what you can see is in the non-surrounded nucleolus stage in particular, you see an increase in the size. So we think it's actually truly decondensed enough to even push out the nuclear size. And the other interesting thing we see is usually what happens is oocytes during that chromatin reorganization go from a couple of nucleolar-like bodies to a single one. In wild type, you almost have hardly any of these, but in Nemp mutants, you can see there's a bunch with 2 nucleolar-like bodies, and sometimes we have 3 or 4. So really dramatic changes in chromatin. Okay, so what's going on when we're seeing these changes in chromatin? So we went back with our beautiful antibody and started looking more closely and started doing super resolution microscopy. And when we did, we got a real surprise that is currently one of my fascinations. So what you can see here is that this is a primordial oocyte, and the Nemp staining is, as I showed you before, very ubiquitously smooth around the inner nuclear membrane. But then as it starts to grow, all of the Nemp, or I don't know, all the Nemp, the majority of the Nemp everything we can stain seems to be reorganized and becomes little islands inside the nuclear envelope, scattered through in a remarkably evenly spaced size and shape and number. So my postdoc Abira has been doing a lot of microscopy and quantitating it. What you can see here is as the oocyte grows, you can see that it starts to have a large number of these, reproducible number of these dots. We get up to more than 100 in every single oocyte at that stage. And the size is truly remarkable. So these are huge cells, but you can see here that the size of these oocytes, of these clusters in the secondary oocyte is more than half a micron. And so now we're really fascinated with knowing what's going on with these Nemp dots. What are they doing? So we've made a Nemp GFP mouse, so into the Rosa locus, and then you can do a lock-stop-lock, so you can express it wherever you want, when you want to. And so we use a germline specific CRE to drive Nemp expression in a Nemp mutant animal. So first of all, there is fertility rescue. It's not 100%, but you do get offspring again. And we've done it with ZP3 CRE, which comes on a little bit later in the ovary. So we're also doing a FOSA to see if that's what it is, or is there a little bit of problem with the GFP added to it. We're doing proteomics. We just gave it off to Ben Major today, I think. And we're also doing live imaging. And what's really cool is that, so this is actually, so first of all, that Nemp goes into these dots. And you can see that, so this is Nemp GFP in an oocyte that we're doing live imaging on. And so this nuclear envelope is going back and forth, and this Nemp is sort of staying in these clusters. We're doing photo bleaching experiments. We're trying to understand better what this is doing. So do they participate in chromatin conversation? What are its other binding partners? And so, okay. So yeah, so the females, so mice that lack NEMP1, the females are infertile, they have altered chromatin organization, they increase number of, I didn't say this, but they have more of those premature oocytes, non-surrounded nucleolus, and an increased nuclear size. And they form these like remarkable clusters that are obsessively. So obviously, is this something that's conserved? So I'm super excited because we've just started this collaboration with the in vitro fertilization clinic at WashU. And for the last 2 or 3 weeks, my postdoc gets a phone call, he runs over, he gets the oocyte. So basically when women are undergoing fertility treatment, immature oocytes are thrown away usually. So if there's a consent form, then we're allowed to take them, and there's a history of them. And so it's unbelievable, but this is a human oocyte and this is NEMP1 staining. So we are really beyond thrilled with this. It looks like there's more. We've counted them, there's more. We don't actually know why, if this is age different or species different, but it does suggest that these clusters are likely functionally important since they're conserved. Do I have time to tell the other little story? Am I done? Because it seemed like we took a little while to get started. It's time? You sure? Okay. So a quick question would be, what else does NEMP do? And what we noticed, I'll go really quickly through it, is the NEMP mutants, even though they're viable, they have a huge splenomegaly. Okay, so why would that be? So, and that's just quantification. So you get splenomegaly sometimes because it's a secondary site of blood production, and if you have anemia. And so we did quantitation of the number of red blood cells in NEMP mutants, and they were reduced, and the blood cells were odd looking. So what's super interesting about blood cells is they do radical stuff with their nucleus and their chromatin. So when an erythroblast becomes a red blood cell, it shrinks its nucleus down from this, mostly the cell size to this little tiny pellet. It pushes it to the side, it polarizes, and then it spits it out. And that's how you end up with a red blood cell that is able to effectively transport oxygen. So Nemp is expressed in these cells. And what was interesting, so we went back into the blood, we did FACS with Casey Choi at WashU. And so basically they go from pro-E to area A, area B, area C, and then they extrude the nucleus. The blue is the het, the wild type, and then the red is the mutant. And what you can see is there's actually more progenitors for the red blood cells at the area A stage in the bone marrow. But quickly, when it gets to the area B stage, they drop, they're disappearing. And so if we go back and we do an exon staining to see if there's death, what we can see is there's not much death early on. So if you just look at the red, but basically when you get to that area B stage, the Nemp mutants, the erythroblasts are dying as they're undergoing this chromatin condensation, or they're attempting to do chromatin condensation and enucleation. And then what's super interesting about blood cell, which you may not know because I didn't know, blood cells go through this remarkable way of dealing with this structural change in their chromatin. In that there was this cool cell paper from Peng Ji's lab a couple of years ago, where he showed that as these cells are losing all these histones, shrinking their nucleus down, they actually open up their nuclear envelope, spit out the histones, close it again, open it up, and they go through several cycles of it. It's really remarkable biology. I was super excited about that. We went and talked to Peng and started collaboration with him. This is just Nemp staining to show you that there's Nemp, and then this is the extruded chromatin. This is the Nemp. You can see that it's surrounding that hole. And so what Peng had shown before is this is just lamin B staining of these erythroblasts as they're undergoing this process. And you can see these little half moons, right, where the opening up of this nuclear envelope. It's a crazy process. But in the Nemp mutants, what we see is we have a substantial and significant decrease in the number of nuclear openings we can detect. And so currently we're trying to understand, what is it about Nemp that is important for this nuclear opening? And we have two completely different hypotheses. So one hypothesis is that Nemp is needed to kind of open it up, right? Or maybe close it down again. Another hypothesis, which I think is maybe more trivial, but still interesting, is that maybe without Nemp to support the nuclear envelope, once you do the opening, you can't hold it together enough, right? So it's lost this stiff corset or something that allows you to close again after the histone release. But that's kind of where we are with this. So aside from its role, its obviously conserved role in germ cell, it also is important for red blood cell development. It supports I believe it supports the nuclear envelope of the erythroblast during nuclear envelope opening, and it either lacks opening or we can't find, once it has it, they're disappearing. So those are kind of our models at the moment. And then I just want to thank the people who did this. First of all, it should have been bolded. This started in Canada with my fabulous friends and collaborators who were so generous in taking this animal odyssey. So Andrea Jusakova is an oocyte expert, and so she's been a great collaborator, and we continue to collaborate with her. So with the early studies, we had had no fertility from the animals for 6 months, and I was like, oh, it doesn't mean anything yet. So she really came in and helped us move it to another level. Anne-Claude Gingras is the person we did the proteomics with, which was really great. Brian Saruna and his student Curtis Boswell, they knocked out Nemp1 and Nemp2 in fish. Lawrence Peltier helped us with the super resolution microscopy, and Zhu Sun and Qian did the devised this special AFM and helped us with that. And Yanit in my lab did so much of the work I can't even tell you, and Robin is the grad student who made the first knockout of Nemp and showed that it was sterile in Drosophila and characterized a lot of the phenotypes. And now down in the US, now that we're here, the blood work was done in collaboration with Casey Choi and Peng Ji in Chicago. The FM, where I was showing you that plated cells can now survive on soft substrates, and was done with Amid Pathik. And the worm knockout was done with Tim Shadle. Is that it? Oh yeah, and then these are the people in my lab. The yellow are the people who are currently working on trying to understand what Nemp is doing and these dots and everything. And yeah, that's it. Thank you. I'm happy to answer any questions.
I have two probably dumb questions, for all honest people aren't thinking of better ones, but I couldn't put together two things. One is on the LOX mutants, the stiffness. I mean, if I think of ovary or erythrocytes, those are not in the stiffest environments in the body that I can think of. And I'm surprised, regardless of how stiff the environment is, this would affect the nucleus in a specific way. I might smash the cell, but I don't think it would necessarily impact the nucleus. So I was wondering.
I mean, I know we're trying to start. So first of all, I guess just to back up, I think the fact that the plating on different substrates and reducing cross-linking, and actually I didn't mention it, but it's very preliminary now. I think we've only done it with 2 ovaries, but in a LOX mutant, we saw a softening with the AFM.
Yeah.
Or Amid. I guess, I mean, I'm struggling to understand that too. My thought is that maybe it's mechanical pressures that are transmitted through cytoskeletal elements that go in.
And then I think you said at the beginning, maybe I just didn't keep up with this, but Nemp is expressed in all cells.
So in flies, it's expressed in all cells. In mice, Nemp1 is strong in the germline. Nemp2 is in the stroma.
I see. And is human similar? Because the cell lines you're using aren't just ovary.
Sorry, I was speaking just about the ovary then. So for most tissues, Nemp1 and Nemp2 are kind of often co-expressed, and sometimes Nemp1 is expressed in more places. But there's not a simple. But just to jump to it, we've also made Nemp2 mutants. They have no phenotype. We've made Nemp1 mutants. And so they're alive. So it's weird but true. That's science.
So do you think that are there compensatory genes that look the same?
Yeah, so that's actually we definitely think that there might that look the same, not overtly. You can search with DUF2215 and you basically get this, but I do wonder if there's some compensatory gene expression, because in fact, if we knock down Nemp in some tissues in the fly, we can get a stronger phenotype than if we do transient knockdown and if we do long-term knockdown. So we think that maybe the system is compensating itself.
One thing was looked at changes in gene expression.
Mm-hmm.
So you see effects on chromatin, and are the phenotypes that you see with Nemp due to the changes in gene expression because of chromatin reorganization?
Yeah, so of course we looked at gene expression. We've done RNA-seq of Drosophila larval testes from nulls, where we've knocked it down just in the germline. We've done GVs, oocytes from the mouse. We see genes change. We can validate their change. But, there's no gene yet that I can say that's the one that's causing the defect. You know what I mean? We do see changes in transposons with a massive expression of transposons, but different ones in different tissues. We've done RNA-seq and ATAC-seq in wings. So we've got a lot of data, but whether the chromatin changes are just sort of generally messing things up, or if they're more specific, we're still trying to figure that out.
What about things like 3C, when you're looking at what is that?
So super interesting. So I didn't talk about it because there's not enough time, but about a year ago, there was a paper published in, I think it was Microsoft, it might have been DevCell, where they showed that oocytes, as you go through this activation of and changes in chromatin, they get a very unique chromatin structure, which they call pads and iPads. And what happens, instead of the normal big pads, there's lots and lots of little close cross connections. So when they do their, TAD diagrams, and you have that, you get tons in oocytes, and you don't get it anywhere else. So it's a very oocyte specific, and it appears at the same time as these Nemp dots. So like, I'm super excited about the idea that Nemp is either directing it or corralling it or something. So we're doing, so to finish my meandering sentence, Ting Wang we started collaboration with Ting Wang, who's in genetics at WashU, and we've gone 3 rounds of single oocyte high C. It looks like we're getting in wild type, something that looks like what was published, and so we're just about to try it with their help in the mutants, because I think that's an obviously exciting possibility, yeah.
So following up on Dee's question, it sounds like you probably won't know this, but because it does seem like in terms of linking tissue stiffness to nuclear stiffness, there's likely got to be a connection with cytoskeleton. I was struck by your live imaging and kind of what looked like the stability of at least where those dots are.
Yeah.
And so I'm wondering from your proteomics yet, have you gotten anything that links, like, are there associations with sun domain proteins?
Yes. We get sun domain proteins. The other thing too.
And I just wonder, like, can you perturb the cytoskeleton and just ask what happens and start getting at it?
Absolutely. I mean, I think so. If we want to go to my true hypothesis, everyone's excited about intrinsically disordered domains. Nemp has 2 of them. So, like, to me, it's screaming out that there's a phase transition going on as it's going through stage. It also has consensus phosphorylation sites that are affected in apoptosis. So, you know, we've made mice now that lack that nucleus phosphorylation, but we haven't characterized them yet. In terms of the cytoskeleton, you know, so in the knockdown cells, which we've only done thoroughly so now on stiff substrates, so we have to now go back on soft substrates and do the analysis again. We do see some changes in the active cytoskeleton phosphorylation, but we're not really sure. But yeah, that's obviously really exciting ways to go.
I was curious because you talk about the testes
Yes.
in flies, but then not in the other, in the other
Yes.
system.
Oh,
So,
right. That's
and I didn't know whether that was because of the differences in expression, but whether because right, is there something interesting about it being in germ cells and flies, or is that, right, is there something functionally relevant?
yeah. So first of all, the reason that it's just functional because it's an X-linked male sterile. So it took us a long time to be able to get the females. And so we did all of our initial analysis on the males. And then the other dirty secret, I guess, you know, it's not dirty, we said it in the thing, but Nemp1 mutant males are fertile in mice,
Right.
but not in fish or
Okay, that's what I was asking,
or
'cause-
flies. Like we
But
don't.
is it because of the expression pattern that is different, right?
No, it's
Oh.
so Nemp1 is expressed in the testes. You actually, you can see kind of during snapping complex, you can see stain. You can see it all the way through. But the only thing is when we've done sections of mouse testes, we still see some residual staining, and it's the only tissue that we don't see in right. So if you look at ovaries, I showed you the Nemp is gone, right, in our mutants. But if we look at this exon 3 deletion in the males, we still see some staining. So I'm not sure if there's some weird alternate splicing, or is that just a weird background? So we're actually remaking a new allele to see if we can get around it. Because I guess I suspect that it's got to have something to do with male fertility too. But
Yeah, no, that's interesting.
yeah.
And then what happens to Emmerich when the dots form? Is it?
So we don't see Emrin at the dots, but Abira and my postdoc is very interested in what she believes. So we've done a lot of analysis of, so now we have this great mouse where we can make Nemp GFP. And so we're trying to see if it is a phase transition. So in heterozygotes, you seem to have less of the dots a little bit. When you overexpress Nemp in a wild type background, you start getting some dots in the primordials, like what you never see wild type. In Emrin mutants, she sees it being more variable, the dots. And so she's trying to quantitate that now. So, but you know, we don't know if that is that a residual of the early expression, or is there just lower level expression of ubiquitous they're not seeing. So Emrin mutant mice lose their primordial reserve as well, but they still stay fertile. So, and you know, we think Nemp is acting at a couple of different stages. So that's kind of a meandering answer, but that's what we got.
Fascinating. Yeah. I think I don't know about the software, but I know that there's histone specific. There's histones that are specific to the oocyte. I'm wondering if the appearance of these spots of them around the oocyte are related to histone replacement that might be perhaps microdomains analogous to the histone recycling that
That's a super interesting idea, which I haven't had before. So yeah, that's a super cool idea. We've looked at histone staining and a ton of histones. And so generally speaking, we see like an expansion that I think is due to the expanded chromatin, but we do see loss of HPK9 level 2, HP1, and really weirdly, gamma H2AX is reduced in these sites. So something is going on with the histones, I think, and we need to get Melissa to help us figure out what that is.
Along those lines, I guess, I know that some nuclear envelope proteins, especially include forex, have been directly implicated in helping to promote heterochromatin formation. Do you think that these proteins might play an active role in coordinating
That's
compaction, or is it more of a passive role in the loss of structure?
This is like, endless conversations in the lab. Like, so first of all, we have done work in Drosophila S2 cells with DAM-ID, and what we see is that Nemp labels swap, like kind of like lab, akin to labs, it seems to label chunks of genome. And we did labs at the lemon at the same time, it's not the same one. So that's, I guess, interesting. So we're doing live imaging with these oocytes now. And so, yeah, some people in my lab, I'm not convinced, but some people think they see spools of chromatin being, and we just don't, we need to improve our imaging a bit more. But yeah, that's one of our fun ideas for sure, that it could be like really